Hello everybody, we are going to start talking about blood, chapter 19. And as you know from before, it's a fluid connective tissue and it contains both red blood cells, white blood cells, as well as platelets or thrombocytes. It's important to understand in this chapter that we are actually going to be doing a blood typing lab. So the kit that you purchased from the bookstore contains a special kit so that you can actually perform blood typing, and that's really cool. Everybody always likes blood typing lab, especially if you don't already know your blood type, so that's a fun one. So make sure you have your blood kit already ordered and received, because that lab will be coming up shortly. Blood has several functions and key characteristics. One of the most important characteristics that blood has is it is a very tight pH from 7.35 to 7.45. If you go above or below that, you can get into alkalosis or acidosis. So there are buffers within your blood to make sure that your blood stays in a really narrow, tight pH. Blood is also a little bit warmer than if you were to take your temperature. Usually we are 98.6 or 37 degrees Celsius, but blood actually runs a whole degree warmer than our usual body temperature. So it runs at 38 degrees centigrade or 100.4 degrees Fahrenheit. Blood can be and it is 7% of your body weight. So if you wanted to take your body weight and put it into kilograms, so 2.2 pounds per kilogram, and then you multiply your weight in kilograms times 7%, you can figure out how much blood that you have within your body. So I just put a sample calculation in your lecture notes. You should know how to do that. The other thing is that males generally have more blood volume than females, and that's because testosterone actually promotes the production of blood. So males tend to have more blood in their body. The other thing that we need to talk about is the major functions of blood. Really, I think everybody knows that red blood cells carry oxygen or carbon dioxide, depending on whether you're bringing oxygen in, taking carbon dioxide out. Blood is a key component in regulation body temperature. Blood is also key in that it protects against infections, pathogens. So those are our white blood cells that we're thinking about there. Also, our platelets or thrombocytes are involved in clotting. So I've listed all of the key functions that you need to know in your lecture notes. So we've all been to the doctor before to get our blood drawn, or I think most of us have anyway, or we work in the healthcare industry. So this picture should look somewhat familiar to all of us. So if we need to take a blood test, usually we do this for diagnostic reasons, or to see if we're just healthy, maybe check cholesterol, whatever the case may be. So what we do is we draw out whole blood. But then we want to separate that blood into its component parts. So this is called fractionation. And the way we fractionate something is to put it in a centrifuge. We spin it at a really high rate, and that separates the whole blood into its component parts, which are plasma and then what is called the formed elements. And the formed elements make up or is composed of your cells, both red blood cells, white blood cells, as well as your platelets, which are considered cell fragments. If we look at just plasma, this is really the majority of our blood separates out into plasma, about 55%. And this is actually mostly water. So it's the fluid part of the blood. It contains different plasma proteins, nutrients, electrolytes, so on and so forth. And it actually has the composition of interstitial fluid. So it's important to understand that it's very like that tissue fluid or the interstitial fluid between our tissues and cells. The buffy coat is part of your formed elements. So when you spin this tube down, you dropped out the formed elements. The majority of your formed elements is your red blood cells, about 45%, but you have this really, really tiny part called the buffy coat, and this is your white blood cells and your platelets. Remember though, that is still part of your formed elements, even though it forms this buffy coat or this third layer within the test tube. This is a great slide showing your red blood cells, which are the disc-like cells, and your white blood cells, which can be very large, your platelets, and then finally that watery plasma, that liquid part of the blood that comes out in your 55%. So next we're going to look at our plasma proteins, and when we look at these, the biggest one, of course, you can see here on the pie chart is your albumins. This makes up about 60% of your plasma proteins, and they really contribute to your osmolarity and your osmotic pressure within the blood system, and they also are responsible for transporting things like fatty acids and thyroid hormones. The next major group that you're going to see are your immunoglobulins or your antibodies. And the IgG on the chart, you can see IgG, IgA, IgM. These are all part of your globulins. And they are going to be responsible for transporting a number of different items, such as hormone-binding proteins. They are going to transport things like iron ions, lipoproteins, such as lipids and triglycerides, as well as things like testosterone. So your antibody or globulins or immunoglobulins are going to transport lots of different types of things within your blood. Then we get to fibrinogen. Fibrinogen only makes up about 4%, but it is key to the clotting mechanism within your blood. So it's a soluble protein that functions in blood clotting. And fibrinogen is what gets converted into fibrin and leaves the serum part of blood or the fluid part of a blood sample. And so it's important to note that when you take blood, you have to actually indicate, is it plasma or is it serum? And the reason is because serum has all the clotting factors removed from it versus plasma protein. You know, if you were to take the plasma, it's still going to have clotting factors in it. But as soon as fibrinogen converts over to fibrin, it is going to be without the clotting factors. So just something to keep in mind. Then you have a whole series of other plasma proteins. They only constitute about 1% of the entire field of plasma proteins. So we're not going to go through and name those individually. So all those plasma proteins we just talked about are all made, or 90% of them are made within the liver. So if anything goes wrong with your liver cancer, cirrhosis of the liver, it can actually change the composition of your blood because it's responsible for making these plasma proteins. Then the plasma cells themselves are responsible for making antibodies, and then your peptide hormones, which are carried within the blood, are actually made in your endocrine organs. When you start looking at blood cells, formed elements, the first time a student puts a slide on the microscope, they'll see all these purple discs, and they'll be like, I don't see anything. And what they're really looking at, those discs, even though they're purple, that's because of the staining process, are actually your red blood cells. Those little teeny tiny, very light colored fragments, those are actually your platelets. And there's actually a few, if you looked in here, you can see a couple of your white blood cells. So when you're looking at a slide, all those little purple dots, they are actually red blood cells. And if you're looking at those on scanning, you really won't see much of anything, so you have to go up a few powers in order to really differentiate your red blood cells from your white blood cells and your platelets. One thing that's important to understand is formed elements includes all of your cell types and your platelets, not just your red blood cells. I have had that on tests where a student will answer, oh yeah, that's just the red blood cells. Well, no, even though there's not a lot compared to the red blood cells, you have to remember all the cells together. Now, let's talk about hematopoiesis or hemopoiesis. They're the same two terms that mean the same thing, and this is the production of your formed elements, so all of those cells together. We usually talk a lot about red blood cells first, and then we go in and talk about white blood cells. Remember, red blood cells, the reason that that's what you predominantly see is because they make up 45% of all your blood. And if you were to look at all the cells in your body, every one of them, blood cells, red blood cells specifically, make up a third of all of your cells, so 33%, and that's pretty amazing. If you looked at just the formed elements, the red blood cells make up 99.9% of all the formed elements. So they definitely are the winners when you're going to look at blood. And of course, we know already that red blood cells contain hemoglobin. You can calculate how much red blood cells are in you because you can actually do a red blood cell count. You can do this on a slide. You can also determine the amount of packed cells or red blood cells if we want to take what is called a hematocrit. And when you put the blood on a centrifuge, the formed elements come out of the suspension, and then you can take the percent or volume percent to find out about the red blood cells specifically within the blood. Now, if you take a hematocrit, usually this is on a machine, and if we were in the lab, we would be doing this on a machine, but we can also measure it within a capillary tube. We can measure it with a special plate, or we can take and measure it with a ruler. So we'll be doing that in lab. Now, hematocrit, of course, we talked about this is the percent volume of the red blood cells. So why would we take a hematocrit? Who cares how many red blood cells we have? Well, it can be really important to diagnosing any type of problem. If you have internal bleeding, your hematocrit could be unusually low. Or if you're having problems actually producing red blood cells or forming red blood cells, you may have a very low hematocrit. If we take a closer look at the red blood cells, they form these stacks of, almost like a stack of coins, and these are called rouleaux. So the reason that red blood cells form stacks like this is so that they can actually fit through narrow blood vessels. So you can see they're kind of stacked up and going through these section capillaries. Another important thing to remember about red blood cells is they do not have a nucleus. So when you're looking at, or if you were asked a question, a blood cell with a nucleus, you would not say a red blood cell. So that would be one of your key points there, no nucleus. One of the things that's really important to understand about the structure is these bioconcave discs, not only can they stack up like coins, but they have a very large surface to area ratio. And this is important because it enables them to pick up and release oxygen very, very quickly. This is key to getting oxygen throughout the rest of our body. The other thing that makes the shape of the red blood cells important is they are able to actually flex or bend or kind of reform themselves a little bit to fit through tight spaces when they enter, really small capillaries. So I included in your lecture notes, you can have a 7.8 micrometer diameter red blood cell can actually fit into a 4 micrometer capillary. So that's pretty important because that's about half the size, and they're still able to fit and accommodate that really small space. If we are looking at red blood cells, it's important to note that they only live about 120 days. They are not able to divide because they don't have a nucleus. So not having a nucleus means they can't divide, they can't produce proteins, and they also can't repair themselves. So once a red blood cell is out there, it lives a fairly short lifespan because it's not able to reproduce itself. So red blood cells contain hemoglobin, and hemoglobin is a quaternary protein. You've seen this in your chemistry classes, of course. And the important thing here is to note that there are 4 heme groups that all carry iron, and this is important because iron likes to associate with oxygen. So it gives it the ability to carry that oxygen to different parts of the body. That iron, if it actually is holding onto an oxygen, it's called oxyhemoglobin. If it lets go of that oxygen, it is called oxyhemoglobin. If you look at the functions of hemoglobin, of course we think to carry oxygen, but it can also carry carbon dioxide. And this is important because it depends on what you're doing. You're bringing oxygen to the tissues, but the tissues are getting rid of carbon dioxide. So how do they do that? Well, they hand over that carbon dioxide to the hemoglobin, and the hemoglobin becomes caramino hemoglobin, and that is when it's binding to carbon dioxide to take it to the respiratory system to get rid of it or to form different products with the carbon dioxide. So if the oxygen is low, hemoglobin releases oxygen, and then it binds to carbon dioxide. At the lungs, where oxygen tends to be high, then hemoglobin is going to bind to the oxygen and release carbon dioxide. So depending on where you're at in the body will determine if hemoglobin is going to bind to oxygen or release oxygen. If we don't have enough hemoglobin, so we take our measurements of hemoglobin in grams per deciliter, and this is going to tell us whether or not we have enough oxygen carrying capacity within our body. If we don't, if our hemoglobin or possibly our hematocrit runs low, then that would mean that we might have what is called anemia. And there are several different forms of anemia, but one of the things to remember, if you have low iron, a lot of times you will be considered anemic, and that's because you don't have the oxygen carrying capacity within your body. You might feel weak, really tired. It can affect the brain. You won't be allowed to give blood. That's actually how most people find out they're anemic, is they go to give blood and they're like, no, you've got low iron, you are anemic. So as we're looking at hemoglobin, and within the body, red blood cells carry the hemoglobin, and these red blood cells are going to reproduce and form new red blood cells constantly. Every single second, about 3 million new red blood cells, and of course all these red blood cells are going to be carrying that hemoglobin, which is going to carry oxygen or carbon dioxide throughout the body. So we get new red blood cells through a process called erythropoiesis, and it goes through several stages. It's different from an embryo to an adult. In an adult, we get our red blood cells forming in our red bone marrow, and this is also referred to as myeloid tissue. And we start out with hemocytoblast, and these are actually going to be the stem cells that divide into 2 types of tissue. They could become myeloid stem cells or lymphoid stem cells. Now, what type of stem cell it is going to determine what type of blood cell actually gets produced. So let's just look at erythropoiesis. So that means we're only looking at the red blood cell here first. We start out in the red bone marrow. On day 1, we have a pro-erythroblast. So pro kind of means before. And then we go from that to an actual erythroblast. And if you notice on day 3, you get the polychromatic philic erythroblast and then a normoblast on day 4. Now, what's important about day 4 is that's when you have the nucleus disappears. Okay, so we actually eject the nucleus out of the red blood cell. Now, this is the term I want you to remember, reticulocyte. So the reticulocyte is what actually enters the bloodstream and becomes the mature red blood cell. So there's lots of names here and you're like, oh my gosh, how am I going to remember these names? What I want you to remember is erythropoiesis happens in the red bone marrow. It starts with a proerythroblast and it finishes with a reticulocyte. The reticulocyte does not have the nucleus, and that is what forms into your mature red blood cell that enters the bloodstream. So don't get hung up on the names. Remember, reticulocyte, that's what's going to turn into your mature red blood cell. So we make new red blood cells in our body in response to low oxygen levels within our tissues or somewhere in our body. And this is caused by the hormone called EPO or erythropoietin. And in order to start making new red blood cells, we need certain minerals or certain components in order to make the red blood cells. One of the big ones is iron, and the iron is actually transported in the bloodstream by transferrin, and that is going to, of course, be part of the hemoglobin. We also need B12, vitamin B12, vitamin B6, folic acid, as well as amino acids are all required. If you do not get enough vitamin B12, you will actually start getting a type of anemia called pernicious anemia. So it's really important to get vitamin B12, or we're like, well, that shouldn't be hard, right? You just get vitamin B12 in your food. Actually, not always the case. Vitamin B12 can be very hard to absorb after the age of 50. You can also have a hard time getting vitamin B12 if you are vegan. So I actually don't eat much animal foods, so I actually get vitamin B12. I take a supplement every day, and one of the reasons is to make sure I don't get this type of anemia called pernicious anemia. So what happens after the blood cell has run its course? Well, after about 120 days, the red blood cell is exhausted or damaged, and it enters either the spleen, first of all, or if we don't have a spleen, like we had to have a splenectomy, the liver can take over and do the spleen's job. So within the spleen, there are phagocytic cells that are going to engulf the red blood cells. But what they're not going to engulf are the heme units within the hemoglobin, specifically the iron. So the iron is going to be recycled and reused. The rest of the hemoglobin that is not broken down or phagocytized is actually going to leave the body in our urine. So it's going to go through the kidneys and leave through the urine. So there's a couple of key terms that you should recognize when we talk about the recycling of hemoglobin. And the first one is hemoglobinuria, and the second one is hematuria. So why would we have these two terms when we're talking about hemoglobin breakdown? Well, we know that the red blood cells live about 120 days, and then they start to break down. They go through what is called hemolysis. If too much hemolysis is going on, that can actually dump into the urine, and you would have this reddish, brownish colored urine, and we know that there would be too much hemolysis. If we have hematuria, that's when you actually have blood, in whole blood within your urine, and this usually comes from injury to especially a kidney. If you take a really bad hit to the kidneys, you're a fighter, you're a boxer, you're a kickboxer, and you took a really bad punch to the kidneys, you're going to see blood in your urine from that kidney damage. Hopefully it's not too bad and it will go away. Anyway, so as we go on with the hemoglobin that is being recycled, specifically what's being recycled is the iron. And the iron turns into, first of all, it's converted into what is called biliverdin. So there is green in, I think, Spanish as well as French, of course, the V-E-R-T, vert, and so it's going to be a greenish color. Then the biliverdin is going to become bilirubin. And bilirubin is that orange-yellow color. So a lot of times, if you have a baby born with what is called jaundice, or you know somebody who has a liver problem, they become jaundice, they look really orange or yellow. And that means they have a buildup of bilirubin within their body. So not such a good thing. So the bilirubin we saw, of course, was excreted in the liver. Now we look at what happens to that bilirubin. It is further broken down in the large intestine by bacteria. And the bacteria break the bilirubin into urobilins and stercobilins. And some of these are eliminated in the feces, and that's where you get the darker color can be from the bilirubin or the bile going out into the feces. Some of it goes into the bloodstream and can actually go into the kidneys to be further eliminated by the urine. So that is important to note. Now, the other thing that we have to look at is what happens to the iron. We said it was recycled, so where does it go? Well, iron is actually bound to what is called transferrin. And transferrin is a protein that is going to go into the bloodstream to the red bone marrow, and the red blood cells are going to take the iron from the transferrin and use it to make more hemoglobin.